A very good morning to all the delegates, dignitaries, participants, ladies and gentlemen. It's indeed a moment of great pleasure that the Indian National Science Academy Kolkata chapter, jointly in association with IQAC and Unnat Bharat Avian Unit of Kharagpur College, is organizing a national event webinar on body's fight against harmful invaders, how we fight coronavirus and for how long. And myself, Dr. Indranil Chakraborty, welcome you all to the event. We are fortunate enough to have with us Professor Dr. Chandrima Shah, respected President Indian National Science Academy, New Delhi, as the keynote addresser. Our second speaker of today's webinar is Dr. Partha Chakraborty, Principal Scientist, Cell Biology and Physiology Division, CSIR, IICB, Kolkata. Before proceeding further, I'd like to request the audience watching the webinar in YouTube platform to type their queries in the YouTube chat box. We shall communicate these questions to the respective speakers during the question and answer session, which is scheduled to be held after completion of both the speeches and the concluding remarks. And those who are watching in Google Meet platform are also requested to ask the questions during question and answer session. Now, I would like to request Sri Sandeep Sen, respected DSC member, HS Council, to give a brief introduction of today's event.
Good morning, everyone. We are now in AC, that is after Corona, and memorizing the BC or before Corona. Today, the subject of webinar is body's fight against harmful invaders, how to fight coronavirus and how long. Just 1 year ago, in the Corona first wave situation, a national level seminar in this college was held. We conducted Corona awareness program where no vaccine, no medicine were there. Only protection and prevention, combat the evil virus. Though awareness program of INSA and NASI is social responsibilities for the scientists, so they continued the program. In first wave of Corona, younger generation not so much affected, but in the second wave, they are also affected, and third wave knocking the door. In this time, what should we do and what will be? We can learn today from our respected speakers. And in this event also, a competition was held like the former year. Thanks to all.
Thank you, Mr. Sen, for your brief but meaningful introduction. I would now request Dr. Bidyut Samanto, Principal, Kharagpur College, to deliver the welcome address. Sir, are you there? Principal, sir.
Thank you, Dr. Indranil Chakraborty. Actually, I have some problem with video, so I can speak only. Anyway, good morning and welcome everybody to today's national webinar on body's fight against harmful invaders, how we fight coronavirus and for how long. Organized by the Kolkata chapter of Indian National Science Academy, INSA, Internal Quality Assurance Cell IQAC, and the Unnat Bharat Avian UBA unit of Kharagpur College. All of us are passing through the prolonged COVID-19 pandemic situation. After the first phase of the pandemic situation, people adapted new normal situation and are looking for the restoration of the normalcy. Unfortunately, we are facing the second wave of more devastating strain of COVID-19 virus. So once more people are arrested in their houses and we are all in distress. Our students are affected most adversely, although they are now habituated with online class and online examination systems. All the students are eager to come back to their most sacred place, their classrooms and laboratories. But we don't know when we can see our beloved students in their classrooms. All of us are in anxiety. Doctors are apprehending of third wave of COVID infection and are pleading for 100% vaccination. However, most of the different age groups of our large Indian population, including our students, are out of the scope of getting vaccine. And a section of the people are still not willing to take vaccine. In this situation, people, especially doctors, scientists, and government, are worried and trying to find the right path to overcome this situation and how long we are to fight this dreaded COVID-19 virus. Thanks to the Indian National Science Academy, IQAC, and UBA unit of our Kharagpur College to come forward for organizing today's national webinar to make all the stakeholders of our college, neighboring schools and colleges aware of the situation. And a galaxy of distinguished speakers will grace on this occasion. I am proud to say that today we have with us Dr. Chandrima Saha, the present president of the Indian National Science Academy. She was the former director of National Institute of Immunology, New Delhi. She is the fellow of several prestigious academy in the arena of science, such as Third World Academy of Science, National Academy, Academy of Science, and National Academy of Science. We are really lucky and honored to have with us such a distinguished and high-profile person as the honorable speaker. Despite her busy schedule, she will deliver the keynote lecture on germs, how we defend ourselves from harmful invaders. On behalf of Kharagpur College, I welcome you, madam, on this auspicious occasion. We are also delighted to have with us another stalwart and great personality, Dr. Hemanta Kumar Mukherjee, the convener of Kolkata chapter of Indian National Science Academy. He is also the fellow of Third World Academy of Science, National Academy, Academy of Science, National Academy of Science. He is the Senior Scientist Platinum Jubilee Fellow of CSIR in Indian Institute for Clinical Biology. He also involved us to organize a national seminar in a big way in collaboration with the Calcutta chapter of National Science Academy in India, NASI, in July 2020. We are very happy that today also he will address in this webinar. We are grateful to you, sir. We are honored with the presence of another eminent and high-profile scientist, Dr. Chitra Mondal. He is the member of the executive committee of the Kolkata chapter of INSA and fellow of the Third World Academy of Science, National Academy, Academy of Science, National Academy of Science. He is distinguished scientist of Science and Engineering Research Board, SERB, and the scientist of CSIR in Indian Institute for Chemical Biology. Despite her busy schedule, she has spared much time for organizing this webinar and also for organizing a students level webinar competition on 3rd June. She has kindly consented to grace on this auspicious occasion. We are grateful to you, madam. We are also glad to have with us another distinguished speaker, Dr. Partho Chakraborty, MD and PhD. He is the principal scientist of Cell Biology and Physiology Division of CSIR in Indian Institute for Chemical Biology, Kolkata. We are all eagerly waiting to listen from him on the burning issue during the ongoing pandemic situation, deadly corona, how long we have to fight. We are grateful to you, sir, for joining with us in this webinar despite your busy schedule. I welcome you, sir. We are also pleased to welcome another distinguished person, Sir J.C. Bose, Chair Professor of NASI, ICMR NICED, Kolkata chapter, Dr. Amit Ghosh, who is also a member of the prestigious National Academy, Academy of Science, National Academy of Science. He is an ex-director of CSIR IMPCH, Chandigarh. He is kindly consented to deliver concluding remarks on this webinar. We are grateful to you, sir. we must convey our gratitude to Mr. Sandeep Sen, who has already introduced today's event to you. Mr. Sen is the DSC member of the HS Council. He is involved with our college in a big way since last year. He always likes to interact with the students directly. In July 2020, he helped us to organize student level inter-school and inter-college essay writing competition. This year also, he was one of the member of the jury board for student level webinar competition organized by Swachhata Action Plan Group of Kharagpur College in collaboration with NASI. I convey my gratitude to you, sir. Finally, I convey my deep regards to our governing body president, Professor Anjan Chaki, who always inspires us and actively participates in all activities of the college. I also welcome all the governing body members, our morning in charge, respected teachers of our college and other colleges, non-teaching staff, and my dear students. I am thankful to the members of the IQAC UBA unit of our college and the member of the advisory committee, especially Dr. Jyotirmoy Pramanik, Dr. Indranil Chakraborty, and Dr. Subhra Mishra, convener of this webinar, for their untiring effort for organizing this webinar. I am sure that their effort will be successful with the participation of nearly 400 participants from different corners of the country. I welcome all the participants with the hope that all of us will be enlightened and enriched from this webinar about different harmful invaders, including coronavirus, and learn how to defeat coronavirus so that the situation becomes normal and we get back our beloved students soon in their classrooms and laboratories. Be safe and remain connected. Thank you all.
Thanks to our principal, Dr. Bidyut Samanta, for his nice speech. Now, today we have among us the convener of INSA Kolkata chapter, respected Professor Dr. Hemanta Kumar Majumdar, NASI Senior Scientist Platinum Jubilee Fellow, CSIR, Indian Institute of Chemical Biology, Kolkata. I shall take the privilege to invite Professor Majumdar to say a few words for us.
Very good morning, my respected professors, teachers, and beloved students. We are indeed very happy that today we have a very distinguished person, our President of Indian National Science Academy, with us, and she will be delivering a talk after some time. I will say a few words about INSA, because INSA is organized in collaboration with Kharagpur College. INSA is an organization, is an academic organization representative of the government. INSA has been established not in the name of INSA, it has been established in 1935. The then government of India, they felt that there is a necessity of establishing a scientific institution, in fact, who will look after the promotion of science in the country. So, and that idea was mooted in 1930, and later in 1935 it was established. Professor Richard Greger, who was the editor of Nature. Nature is a scientific journal. He visited India. He talked to some of the scientists in India, particularly the editor of Current Science, and they have a discussion with the distinguished scientists in the country. One of them is Professor Meghnad Saha, who was the president at that 23rd session of the Science Congress. I must say that the Science Congress is the father of all the organizations. So it was the Science Congress which was established in India, and that was they were established at the Asiatic Society Bhavan in Calcutta. INSA was also born there with the help of uh, Indian Science Congress Association. Later, various scientists have joined and they have set up a committee, and the committee was looking after the different purpose of this Indian science, of this scientific academy. And then it was decided that a scientific body comprising the various scientists, professors, academicians, learned people should be set up. And it was set up in 1935. It was set up in Calcutta, and then later, eventually it moved to Delhi. There was a great help from Pandit Jawaharlal Nehru, and he was one of the key men for establishing the Indian National Science Academy. This academy has some charter, like they will look after the promotion of science and technology in the country, coordination among the scientific societies, to promote and maintain liaison between science and humanities, and interact with the academies throughout the globe. This is the objective of the INSA, and presently there are something around 1,500 fellows in the whole country. And today we are privileged that we have with us Professor Chandrima Saha, the President of Science, this INSA, who will be delivering a talk. And I thank everybody to be here and to listen to the important lectures by two scientists. One is Chandrima Saha and the other one is very young blood, Dr. Partha Chakraborty, a doctor as well as a scientist. So I thank you very much. We'll start the session soon. Thank you.
Thank you, Professor Mojumdar. In your very small speech, you have summarized the brief history of development of Indian National Science Academy, its glorious past, and also the objective of Indian National Science Academy. We are highly grateful to you for your speech, sir. I would most humbly request Dr. Chitra Mondal, Honorable Member of the Executive Committee, INSA Kolkata Chapter, and also the Sir Distinguished Fellow, Cancer Biology and Inflammatory Disorder Division, CSIR, Indian Institute of Chemical Biology, Kolkata, to kindly introduce Professor Chandrima Saha, the keynote addresser of today's webinar.
Thank you, Dr. Chakraborty. Good morning, everybody, to all the respected teachers, students, and all the others dignitary here. And today, as you all know right now, that we have assembled here to listen to very exciting lectures, which will be delivered by two eminent speakers. They will discuss how our body fight against any harmful invader, including coronavirus. And as you know, you will understand that body always win, therefore we survive. And as Dr. Mojumdar was mentioning, that INSA's objective, and accordingly Kolkata chapter of INSA as well as the NASI, we organize regularly several contemporary program, and specially addressed to the student of school and colleges. And last 1 year, usually we go physically to the respective school and colleges, but last 1 year we are organizing only the virtual program. And in this COVID series, this will be our 6th and 7th lecture. So we always invited different speakers so that they can address different aspects of this deadly disease and also the deadly virus. So now it is my privilege, and I am very happy to announce that Professor Chandrima Saha, a famous biologist and, as you know, President of INSA, is with us and kindly will deliver the keynote lecture. So now it is my privilege and honor to introduce Dr. Saha, And of course, she doesn't need any introduction in our scientific community. However, I'm going to do it as many students are present in this webinar. Dr. Chandrima Saha has a brilliant academic career. She is actually the daughter of the city, and she completed all her education from Calcutta University, including PhD. Then she went to USA for her postdoctoral research, and at the Kansas University Medical Center, and subsequently at Biomedical Center of the Population Council at the Rockefeller University, USA. After returning to the country, she joined at National Institute of Immunology, New Delhi as a scientist, and eventually rose to become the director of this institute. She is an elected fellow of all three scientific academies and served this academy in different capacity like sectional president, more importantly, council member of all three academies, and she also served as a vice president, international affair of the Indian National Science Academy, and subsequently become the president of this academy. And I'm proud to announce that she is the first woman president of INSA. And as you know, it has been established in 1935. So it's a after a long time, a woman become a president. And she is also recognized internationally by being an elected fellow of the World Academy of Science, Italy. For her phenomenal contribution, she has been conferred a large number of awards and honors like Shanti Bhatnagar Medal, INSA, Shakuntala Amitchand Award of ICMR. She became prestigious J.C. Bose National Fellow. She was awarded Ranbaxy Science Foundation Award for Basic Science, Om Prakash Bhasin Award, Archana Sharma Memorial Award, Darshan Ranganathan Memorial Award, Chandralekha Hora Memorial Model. And about her research interest, she was always interested around the elucidation of the process that influence the cell death under varying physiological condition in diverse organisms. So you can, we are all eager to listen her with her vast knowledge on this subject. She is going to tell us the germs, how we defend ourselves from harmful invader. With this very short introduction, it is my privilege to welcome Chandrima, Dr. Chandrima Saha, and please, Dr. Saha, deliver your lecture. Thank you very much.
Thank you, Dr. Mondal, for a very, very kind introduction. All distinguished guests and students, it is a pleasure to be here. And I thank Dr. Chitra Mondal and Dr. Hemanta Majumdar for giving me this opportunity to be in this forum. My greetings to all members of the Kharagpur College, including the principal, the faculty, and the students. Today, I'm going to speak about a subject which I am primarily speaking to students. So there will be very lucid presentations, and I seek the indulgence of experts in this field to bear with me. So as you can see, I will now share my slides. Is it okay? Share. Is it visible now? Are the slides visible?
No.
I'm sharing it. So
it's not visible.
Share. Now, can you see it now?
Yeah. Yes.
Yeah, now we can see. Yes.
Yes, it is visible.
Okay. So do you see the full screen mode?
Yeah.
So as you see, my title is How We Defend Ourselves from Germs. And infectious pathogens have been evolving since life on Earth began. And although it is difficult to pinpoint the exact time when the first pathogen emerged, it may be as old as when the first cell came on Earth. So this is a mystery that we have never been able to solve. But what is important is that during human I don't know why it's jumping. Let me go back to. So, as we know about human evolution, that when about 5.8 to 6 million years ago, humans emerged from apes, and the first human ancestor is believed to have walked on two legs. Then, around 300 to 200,000 years ago, man started using tools, and under the African origin paradigm, about 70 to 50,000 years ago, the modern man, the anatomically modern humans, they descended from a population of Homo sapiens and migrated from East Africa years ago, and spread across the southern coast of Asia to Oceania and came, in certain parts of India as well. So the human migration is a very interesting story, but it's a long story. So all along this human migration, the pathogens like bacteria, virus, fungi, and parasites invaded the human populations. And as the population increased, it became even, these diseases became even more prevalent. But the new species evolved because these pathogens replicate much faster than humans. And so due to very frequent mutations, many new species evolved. So now, I don't know why it's jumping. So when we look at the host, that means when humans were invaded by the pathogens, and when we look at the pathogens are always trying to establish infection in the host, and the host is trying to get rid of the pathogen. So these are two counter processes, and evolutionary selective forces acting on these pathways has actually defined our immune system. So that's again, there are many thoughts on this, and what we know that this race against pathogens by the host has actually established the host some of the very elegant defense systems in the host. And while the pathogen also has evolved very elegant systems by which it can actually infect the host. Now, so as human population increased, diseases came by, and there were epidemics, which is a pandemic is actually an epidemic, but when it spreads to different countries, different continents, it's called a pandemic, and it is the geographical distribution which defines the pandemic itself. Now, a widespread endemic disease where a stable number of people are infected is not actually a pandemic. For example, the seasonal influenza is generally excluded as they are limited to large regions of the globe rather than being spread worldwide. So what we are facing now is a pandemic which has spread all over the world. And so with increase in population, increase in travel, the germs were crossing borders very frequently, and we were getting epidemics and pandemics. So now we are in a pandemic which has altered human history altogether. So this infographic actually shows that just the three red arrows that you can see is the worldwide smallpox epidemic is a very famous epidemic that killed many people. Then the bubonic plague or the Black Death also killed many people. The Spanish flu was during 1918 and 1919 was also a pandemic that is to be looked into for lessons learned from there. Now this is a painting by Peter van Halen in the 17th century, and this is a depiction of bubonic plague in a particular city, that Siena in Italy in the 14th century, which he has actually drawn in the 17th century, thinking about that. He was not really a first-hand visitor of this, but as you can see that the paintings and notes from yesteryears are actually valuable documents that we can get information from. So this particular bubonic plague or Black Death swept through Europe with a very high speed, and it was spread by the bite of an infected flea. This is the flea you can see here, and the bacteria is Yersinia pestis, which actually caused severe devastation that is recorded in human history. The fleas are actually found in rats. So within 4 years, because the rats go from one house to another, and so there is frequent spreading of these fleas, and within 4 years, it killed about 1/3 of the population of Europe, more than 25 million people. Now, another pandemic that occurred is the smallpox. The smallpox, many of you were not born when smallpox actually got eliminated. It was a deadly disease where it killed about 30% of those that it infected. But people who were infected could survive, but they had deep scars on their faces and other parts of the body. So 300 million people died from smallpox in the first 3/4 of the 20th century, and was also the third cause of all blindness. Now, once this happened, there were questions in people's mind that there was a thought that there is something extra to the body which is coming in and causing the diseases. So the germ theory is an early concept, and in the Middle Ages, physicians like Ibn Sina, who is a very famous polymath, and he was Persian, and he was one of the most significant physicians at that time. And he floated such concepts that, as I said, that something other than outside the body is invading the body and causing the diseases. Now, a very significant development happened when Antony van Leeuwenhoek, a Dutch draper, actually discovered the microscope. He used to dabble with lenses because he was a draper, and he used to look into the threads of the drapery that he sold. So he tried creating the microscope, and this is a little contraption that he created. You can imagine the microscope from where to where it has gone. So these are the very small lenses, but very high power, because he used liquid glass to design these. And Antony van Leeuwenhoek, other than the drapery, he started looking at nature, and he looked at many moving, components under the microscope. It is not exactly under the microscope, but this is actually the place where he put his specimens in. And he discovered an entirely new world, which was not seen by anyone before he discovered this. Later on, Robert Hooke and others also improved on the microscope, but it was primarily Antony van Leeuwenhoek's discovery that gave the microscope to us. So Louis Pasteur and Robert Hooke are the two scientists that really provided evidence for the germ theory. There are many others, but I'll just mention these two. They were stalwarts. And Pasteur is regarded as one of the fathers of the germ theory of diseases. And his many experiments really showed that diseases could be prevented by killing or stopping germs. And then Robert Koch also was very famous, and he was a German physician and microbiologist, and was the discoverer of specific causative agents of deadly infectious diseases like the tuberculosis, the cholera, and the anthrax. He is regarded as one of the main founders of modern bacteriology. As the germ theory evolved, the way we defend ourselves from germs also started to be in the thoughts of scientists. And there were many questions that came after this. And what the questions were like: why is there sickness? Why only a few people are sick at a time? And why not everyone? Why many people are afflicted sometimes together, like in epidemics? Why some recover better, and why some succumb and some do not? So the question then came, the sort of science of immunology developed from here, And it's a very interesting story. It's looking at looking at our ability to defend ourselves from something that is not a part of us. So the immunity is actually something that we have learned over the years, and many elegant experiments and many elegant observations have gone in understanding immunity. So I will give a very brief introduction to that, in the sense that the first discovery of immune cells was actually made by Eli Mechnikov, who was a Russian scientist. And what he did in a very elegant experiment, he took starfish, and starfish larvae are transparent. So he put a rose thorn in it. And when he put a rose thorn, he saw that cells were actually homing in on the thorn, trying to sort of engulf it and destroy it. So this was the first observation that he made, was of phagocytosis, in the sense eating up of bacteria or eating up of a foreign object. And he studied phagocytosis for a long time, and he published very nice papers, for which he got and for this discovery, he got the Nobel Prize later on. Now, if you look at this sketch, you see that this is actually what happens if an insect bites your skin, and under the skin, this blood bubble form, and the immune cells actually come in. So the immune cells that come in, they try to fight off the germs that has entered. So there are two kinds of, and also I should mention here that there are many routes of entry. There can be insect bites, there can be oral ingestion, there can be like we are having now. It can come through aerosols. So there are many routes of entry of the germs, but wherever it is, we do get the first line of soldiers coming in, who are part of the innate immunity. And as you see that this basophil, eosinophil, neutrophil, monocyte, you can see that in your blood reports when they come in. And so the three very important cells in the innate immunity are the neutrophils, the dendritic cells, and the macrophages. Now, the neutrophil and the macrophages are able to digest these pathogens and do a direct killing. Now, sometimes they fail. And in that case, what happens? Sometimes they fail, and what happens is then they seek the help of the adaptive immune system. The adaptive immune system is the fallback of innate immunity, and here also we have the soldiers who protect ourselves. But the macrophages, what they do is they digest up all the pathogens. So do the dendritic cells, and they present after the digestion, the part of the proteins that they get, they present on their surface, and they talk to T cells. And the T helper lymphocyte now gets activated and tells the B lymphocytes to produce high affinity antibodies. So the B lymphocytes actually get into plasma cells, mature into plasma cells, and they start secreting the high affinity antibodies. Another cell, which is called a cytotoxic T lymphocyte, can actually directly kill the pathogens itself. So there are two aspects of this response. This is a very fast response. The innate immunity happens within few hours, and this happens within days. And the principle of vaccination is through stimulating the B lymphocyte. Now, subsequent to Mechnikov's discovery, the macrophage was worked upon, and the dendritic cell was worked upon, and many important discoveries were made as to how they present the antigens. It's a very complicated story, but I'm going to show you some of the areas that we can think about. If you look at this slide, it shows two beautiful scanning electron micrographs. This is a macrophage with philopodia extending, and it is trying to catch hold of these bacteria. And it's an alveolar macrophage, and these are E. coli bacteria. This was set up to see the process, and this is actually a red blood cell. So they engulf, and if you see near the they're actually getting, these are the streptococcus bacteria which are getting into the macrophages. So when the microscope has actually evolved from Leeuwenhoek's little tiny contraption to such resolutions, then we can actually see life itself. And it is so very interesting. Now, what happens when the pathogen is engulfed by the macrophages? When the pathogen is engulfed, it's put in a compartment which is called a phagosome. This phagosome is very rich in reactive oxygen species, which is actually produced by an enzyme called NADPH oxidase. And this phagosome eventually fuses with the lysosome. The lysosome is a bag of enzymes, and it contains many enzymes which can actually digest the bacteria. It just breaks it down, and some of these waste are thrown out, and some of them are presented on the surface of the macrophages. And when the macrophage presents on the surface, the T cells come in and recognize these antigens. So to mount an immune response, which goes to the B cells and tells them to produce the antibody. Many years ago, we showed one that infected cells would have, these are markers for lysosomes, and these are markers for reactive oxygen species. And if you overlap the two, you'll see that it is actually the lysosomes which are lighting up, which means that the infected cell phagolysosomes, where the pathogens are dwelling, there is a very heavy increase in reactive oxygen species, and that is generated by NADPH oxidase that converts oxygen to superoxide, and then it dismutates to hydrogen peroxide, which actually generates these very reactive radicals in combination with nitric oxide, or uses myeloperoxidase to make hypochlorous acid. So this is a compartment which actually is full of these highly reactive species, which in addition to the enzymes of the lysosome acts on the pathogen. And you would think that why then sometimes the innate immunity fails, but it does fail sometimes. It does fail sometimes. So now, if you look at this picture, you see this is a dendritic cell. This is scanning electron micrograph again, and this is a dendritic cell, and this is a T cell, and the T cell and dendritic cells are actually communicating. This is what can be seen by a scanning electron microscope. But if you look at the molecular level, you will see that the dendritic cell is actually presenting the antigen that I told that the fragments of the pathogens is presented by the dendritic cell in collaboration with the major histocompatibility complex, which is a presenter of the antigens, to the T cell through T cell receptor, along with the co-stimulatory molecules. So this, what you're seeing by the eye, is actually a very complex interaction between the dendritic cell and the T cell. So this is again a very interesting picture, and it's almost like somebody has painted this, but this is actually a macrophage. Macrophages, if you grow in culture, they spread out like this, and these are Streptococcus bacteria, and this is the T cell, which is sitting on the macrophage, waiting for it to engulf the bacteria and present the antigen to it, because it has already come to the macrophage knowing that this macrophage is going to engulf the bacteria. There are a lot of chemokines, etc., secreted for this process. Now, this T cell macrophage and the bacterial interaction then happens. What happens is B cell comes in, and the T cell now talks to the B cell through, again, a very complicated interaction, and tells the B cell to generate the antibody. And this is the principle of vaccination, as I said, is used. So the vaccine was discovered in, I think, around the principle of vaccination is very old, but it was mainly discovered by Edward Jenner in the 17th century. The invention of vaccination actually was a turning point in the war between the microbes and humans. And as you know, that vaccines are very represent the most cost-effective life-saving device in history. And so if we look at history, we see that inoculation with smallpox virus material actually preceded smallpox vaccination that we later on saw, and was one of the most accepted approaches to protect from the disease. Now, what happens is that if the smallpox virus material is taken, which is live material, it may kill it may actually kill the person. So later on, heat killed things were injected. But first, the inoculation, which was found in China about 1000 AD, and in India, Turkey, and probably Africa as well, where pustules, where live virus were there, were taken and injected into people, and that gave immunity to them. I'm sure that a lot of people actually got the infection with this and may have died. But the principle was that they got immune, and they were they could resist the disease. So, but the story, interesting story is here where Edward Jenner in the 18th century actually inoculated a boy, who was the son of his gardener, with cowpox. So cows used to get small pox. So cowpox, he inoculated material from cowpox, and then later on challenged this same boy with smallpox virus, and the boy did not get sick. So he was lucky, and but then he started doing this more often, and he accumulated some data by which he is now actually he is known as the father of vaccination by this method, although he actually at that point of time, today one wouldn't be able to do this because of the regulations that we have. So he faced the initial resistance from the medical community, but he increased his vaccination efforts. And as I said, he became celebrated as the father of vaccination. So the question is, how did inoculation generate immunity, particularly an immunity that lasts? So there were many questions on this, and some factor produced in the body must be able to counter the infection, also retain a memory of the infection over multiple years. So when I talked about B cells, the B cells, when they differentiate into plasma cells generating antibody. They also form memory cells, which are preserved by the body, and later on when the similar infection is there, the memory cells expand, and then they they give the immune response to the body. So these were some questions that came in after the immune system, after Jenner discovered the vaccination, and it started as a regular activity. Now, so we have two things. One is active immunization, one is passive immunization. So active immunization is when natural infection can make you immune, generate antibodies in your body, or cell-mediated immunity like the cytotoxic T cells, or the microorganisms, inactivated microorganisms, as Jenner's experiment did. He used a different virus, but killed viruses may be used, killed bacteria may be used. Then there are DNA vaccines now developed, subunit vaccines, like very frequently if you fall, you can be taking the tetanus vaccine. So that's a toxoid vaccine, that's a subunit vaccine. And then recombinant vectors, which are can be bacterial or viral. And so these are active immunization, whereby you are given an agent and the body develops immunity. But there can be passive immunization. For example, the mother to child, there is a transfer of antibodies. So the mother has antibodies, so this is a natural immunity given to the child. But in the artificial transfer, it's like, that now there was attempts to take plasma from people infected with COVID-19 and given to a sick person. And so this was artificial passive immunization, and it can be also done with the recombinant antibodies if you have a process. So these are the two main kinds of immunization. And if we talk about SARS-CoV-2 vaccines, we have to you are very familiar with this figure, I'm sure. But if we recapitulate that this virus has an envelope, and from this envelope is sticking out these spike proteins, and inside is the RNA of the virus. This is an RNA virus. So if you look at this structure, this is a model, that molecular model of the coronavirus spike protein. which is bound to an angiotensin converting enzyme receptor in a human cell. So these ACE2 receptors on our cells are actually responsible for binding to the spike protein of the virus. This is the virus and this is a spike protein. So there is another molecule which facilitates that. It is there to which facilitates the binding of this receptor. But mainly, what we have to, what I wanted to point out here is that this region where the spike protein binds to the ACE2 receptor, the residues that are involved in binding are actually the ones that we would like to have antibodies against, because these antibodies will then be able to stop the spike protein from binding to the ACE2 receptor. So this is a very important thing, and all the vaccines that you hear about this against spike protein, but they can be against some of the structural spike protein, but the most important one are the residues that we find where it binds to the ACE2 receptor. Yes. So these are the different kinds of vaccines that we have. One is the viral vectors, where this is an adenovirus vector. These are regular the adenoviruses that are used where they're made replication deficient, and they are the DNA of the spike protein is inserted into there, and there are two examples. One is Johnson & Johnson vaccine, and another is Oxford AstraZeneca vaccine, where the DNA that goes into the cell, and then I will talk about the mechanism later. And then there is RNA vaccine. RNA vaccine is where they are in lipid particles, and they're given like the Pfizer BioNTech vaccine, the Moderna vaccine, or the Inovio vaccine. So this is a DNA vaccine, the Inovio. So the protein and the proteins or DNA can be attached to nanoparticles, and Novavax is one such vaccine that is coming up. And all the proteins, the subunit proteins fragments are used to make vaccines also. And the examples are Flinders University vaccine. And otherwise, this is the traditional method, which is Covaxin that we are having in India, which are killed or attenuated virus. And this have to be done very carefully because you do not want any live virus to be left in your preparation. So this is made by Bharat Biotech in India. So this is the mechanism how RNA vaccines work. They are in lipid nanoparticles, and then you have the RNA coding for the spike protein, the areas that I pointed out, and they are injected into the human cell, and then protein translation. This is a very familiar figure that they go to attach to the ribosome, and the protein translation happens, and the antigens are expressed on the cell surface, from where, as I told, that the T cells and B cells get activated by recognizing them on the cell surface, like what was being presented by the macrophages and the dendritic cell. So this is the principle of the RNA vaccine. Now this is the DNA vaccine, which is the AstraZeneca vaccine, where you have the DNA coded inside the adenovirus genome, and the adenovirus gets into the cell. It injects its DNA into the nucleus, and then again through process of transcription and translation, the protein is formed. The protein is expressed on the surface, recognized by the T and B cells, and antibody is generated, and cell-mediated immunity as well. So in India, the current ignore the rupees here because these keep on varying. So there is the Covishield vaccine, which is the adenovirus vector vaccine that we have in India and very widely used. Then Covaxin, which is the inactivated virus, and now we have the Sputnik, which has arrived. It is also an adenovirus vaccine that has come in, and there are a lot more being developed now. So one of the important things that are with vaccines is that you have to have herd immunity to actually reduce the incidences of disease. So herd immunity is actually the protection where a large proportion of a given population acquires this immunity. By the processes that I said, they should acquire immunity, and individual variations are there. But majority of the people, if they are vaccinated, they will acquire immunity and can no longer get infected or transmit infection. And this can happen through vaccination or immunity resulting from previous infection. But to get a herd immunity through natural infection is a very tall order. So through vaccination is also a tall order because we have a huge population and we need about 70 to 80% of people to get vaccinated to get herd immunity. Now, this is the principle of herd immunity, where you see that these are COVID-19 infected people, and the blue you see are vaccinated or immunized, and these are susceptible people. So when a COVID-19 infected person transmits virus to an infected person, to a protected person, there is no spread of the disease. But when it comes to the susceptible people, there is a spread of the disease. So this is how herd immunity works. But if you look at this this schematic, you will see that R naught is actually how many people you are releasing as infected. So if there are no vaccination and you release 2 infected people in a group of people, what you find is that it is gradually spreading, and in some time later, it infects the whole population. But if there's 75% people are vaccinated, you will see that introduction of 2 people will not matter too much, and there will be some infections. If you have 5 infected people, then this will change, and there will be a little more infections because you have more susceptible people here. But if you do not have susceptible people and you're protected with vaccine, then the scenario of having most of the people infected will be much less, and you will see infections getting less and less day by day. Now, this is last year's picture, where this is the herd immunity estimate. And if you see New York City, London, Stockholm, Boston, and Madrid, they could not acquire herd immunity like this. I do not have a picture to see it now. This has improved much in the United States. And although the typical herd immunity has been reached, I do not have an idea. Now, the success of vaccines is a very important thing, because a little bit, if you look at a little bit in the history, the smallpox in the 18th century inflicted a lot of deaths in Europe. And in as you go along, in 1977, this smallpox was actually eliminated. And this was eliminated through vaccination, through regular vaccination year after year. When we were young, we used to take smallpox vaccine every year as booster doses. So smallpox got eliminated from the world altogether. So diphtheria also was a problem in the 1940s, but now you rarely hear anybody dying from diphtheria. And pertussis, which is a whooping cough, is again something that was in the 1980s had many cases, but now you do not see much cases of pertussis. Now, this is a very famous picture which shows that Christiana, this is this lady who has been left in this field, and she cannot reach the house where it is, her house, because she cannot walk. The reason she cannot walk is that she has polio. You have heard about polio. Now, polio virus looks somewhat like a coronavirus, but it used to cause many cases. It actually affects the brainstem and thereby causing paralysis. So it created epidemics in the early 1950s, and a lot of children used to get sick. And now it has been eradicated totally from Europe and most of the other countries. And that is again through regular vaccination. You have seen that in the newspapers that get vaccinated for polio, there is an oral vaccine now. So it is much easier to administer. So polio also is getting eradicated like these vaccines. So these are the success stories of vaccines over the years. So now if we look at the coronavirus vaccines, so this came out actually on June 2, this was published. So 95% effective at preventing symptomatic disease, the Pfizer vaccine. Moderna is 86% effective, and 86% again the Johnson & Johnson vaccine, and Oxford AstraZeneca is 76% effective, and 96.4% efficacy against the original virus strains, that is the Novavax vaccine. Now this keeps on varying. Actually, these figures keep on varying depending on the area, the population, the ethnicity, etc. But this was a very recent report. Now, 98% seropositive. There's a publication which is not yet published. It's in the MEDEX review, and 98% seropositivity was found with Covishield in an Indian population of the first line workers in COVID, and 80% seropositivity for Covaxin. This was published. This was actually made available on June 4th. It is not yet reviewed. So there is reasonably good seropositivity, although we do not know what is the functional capacity. So although I couldn't show you, like the last ones I showed you, the success of smallpox vaccine, the success of polio vaccine, but whether these vaccinations are going to be as successful as that, we need some figures, which is still not available. Now, coming to, naturally we come to these variants or the new strains that are formed due to mutations, because they are very rapidly multiplying and we get new strains. And I want to come back to a point that I mentioned earlier, that when they mutate, they form new strains which may have advantages or which may have disadvantages. So this is the one I showed, that there is, in this tussle for the host and the pathogens, there are many mutations that take place. Now, if you look at this very interesting example, is that the glutamic acid at residue 614 was of the virus spike protein was there at the beginning of the infections in 2020, early 2020. By April of 2020, a single mutation in the genome was found, converting the glutamic acid to glycine. And presently, the glutamic acid variant has been all but replaced by the 614G variant. Now, the 614G provides some sort of selective advantage to the virus. So the mutations can be advantageous, can be disadvantageous for the virus. And the virus will keep only those advantageous mutations. So this was an advantageous mutation because it transmitted more efficiently than the glutamic acid, the original strain. And the number of functional spikes on each viral particle was 5 times higher because of this mutation. So naturally, the virus had a great advantage. So the mutants now we are seeing have some advantage or other that is giving them that is giving so much infection. But one thing is there, that the pathogen does not want to kill the host really, because it needs to spread the disease. And so usually the mutations that are kept are the ones that allows maximal transmissibility and maximum replication to the virus. So those are usually kept. And if the host dies, that happens. We are seeing with the coronavirus that the host is dying. But normally they would prefer a mutation that will not cause so much death. So this is what in February 2020 we saw, and this mutation taking over the 614D. And by the end of June, this mutation has all but replaced the earlier one. So this is a very elegant example of how viruses keep the mutations that are advantageous to them. So that I wanted to point out with the first diagram that I showed. I think I'm running short of time. So I really do not want to go into the cytokine storm. This is one example to show that the immune system is not always behaving itself. It can behave in a manner which it can cause this kind of cytokine storm. And cytokines are secreted by the dendritic cells, the macrophages, and it is a part of they are a part of the inflammatory process. And they are produced by several immune cells in the innate system. And 3 of the most important pro-inflammatory cytokines are IL-1, TNF-alpha, and IL-6. Now, IL-6 is used nowadays as a marker for seeing how far the infection has gone. Now, tissue macrophages, mast cells, endothelial epithelial cells are the major source of cytokines. Okay, so the cytokine storm results from a sudden and acute increase in circulating levels of these different pro-inflammatory cytokines. And this cytokine storm you hear many times in the context of COVID-19 infections. So we understand partly, but we do not understand totally that why cytokine storm is occurring in some people and why not in others. and how it can be prevented. So I'm sure the next speaker will allude to this. And you see, this is how it is. These cytokines are secreted by all these cells. And then the T cells are activated. T cells are in turn activating macrophages. So it's a very complicated story that is actually creating uh, organ failures and leading to death. So I thought of putting this. This is actually a scanning micrograph of alveolar macrophage having been infected by, uh, the coronavirus particles here. So we have learned so much from 2020, early 2020 to now, but we have so much left to learn. But it is all I can say is science has really given us uh, many elegant views about life up close. And I think that is one thing that scientists, amongst all this gloom, all this death, the one thing scientists can do is to understand how life works. And we have seen such elegant experiments and such elegant findings in our war with this minuscule pathogen that is trying to defeat us all the time. Thank you very much.
Thank you, Chandrima,
Thank you.
for such an excellent lecture. And even working on immunology for whole year, whole life, I still enjoyed your lecture. It's really good. I'm sure students has thoroughly enjoyed this lecture and so nicely. The slides were so good. So uh, with this thing, I must thank you. And Indranil, please.
Okay. Thank you, Professor Mondal, and thank you, Professor Dr. Chandrima Shah, for your illuminating and wonderfully delivered address. We are really thankful to you, ma'am. Uh, Before going further, I once again want to request the audience, if you have any question, please type it in the YouTube chat box, and we shall communicate it to the speaker during the question answer session, which is scheduled to be held after the completion of our second speech as well as the concluding remarks. Now, our next speaker is Dr. Parthu Chakraborty. I would like to once again request Chitra Mondal ma'am to introduce our next speaker. Ma'am, please.
Thank you, Indranil. And again, my pleasant duty to introduce Dr. Parthu Chakraborty. And he is a scientist at our institute, Indian Institute of Chemical Biology. Of course, he is actually a doctor. He did his MBBS degree from Calcutta Medical College, and then subsequently he completed his MD degree from Benares Hindu University, and then he moved to USA, Boston University of School of Medicine, USA, where he finished his PhD degree, and then he came back to India and joined our institute, that is Indian Institute of Chemical Biology, and working as a principal scientist. He mainly, his research interest is mainly on the diabetic, obesity, and most importantly, he works on metabolic syndrome, which is very, very difficult subject. And in that, he tried to work on non-alcoholic fatty liver disease, cancer metabolism, and etc. And in this journey, he published lot of good paper. And today, being a clinician, come immunologist, come scientist, he is going to discuss on deadly corona and how long we have to fight. Really, Parthu, we are eager to listen to you, that we are tired of waiting. So how long we have to fight against this deadly virus, that we would like to listen from your mouth, and stage is yours. Parthu, please.
Okay. Thank you, Chitradi, for this introduction, and I'm really happy to, have me here. And so after this wonderful presentation from Professor Shaha, I don't have much to talk about. So what I thought that I'll be primarily sharing some of the some of the recent development, particularly in the context of clinical sciences and the biomedical sciences, how it affected us. And I'll try to touch upon some of the aspects of the social concerns, okay, that, okay, social concerns that are related to COVID-19 pandemic, as well as what we have been seeing in terms of confusion and hesitancy in the public domain. So I'll share my slides, and I have some, okay. So I hope, okay. So I hope it's visible to all of you. And okay. So the title of this presentation, what actually I was given about this deadly corona and how long do we have to fight against this, and that's what Chitali also mentioned in the talk. But before I go into the topic itself, I just want to rewind some of the unwanted, unwarranted memories that haunted us for last few months during the second wave of COVID-19. And we all have seen and experienced pain and death. And these pictures of dead bodies buried in the banks of Ganges, and this is pile of fires. People are being burned. People are suffering in front of the hospital, within the ambulance, inside the hospital, and there is a chaos across the country, and it's really gloomy situation we face to it. And honestly, I think we nobody of us, we don't want to see it again, and we should be ready at our levels to address if something happens similar to this in the future. But this, among all this gloom and bad news and these dark pictures, the only silver lining probably we have now is this: that in the September 16 of 2020, we had seen the first peak of the first wave of coronavirus, with close to 1 lakh patients diagnosed every day. And in the second wave, so it reached more than 4 lakhs in May 5th. And now, this is the last data from June 7th. As you can see, the total number of diagnosed cases has went down to 1 lakh. And we have started talking about ending of first wave. But you must remember, this 1 lakh, that cap, this 1 lakh number, is actually was the peak in the first wave. So we are still not done, and all the mathematical models and the epidemiological models the scientists have been putting forward. So it seems that by end of June, we'll come to the baseline as we had experienced in the first 3 months of this year. So that's the only silver lining. But the unfortunate part is that, so we are also, and scientists are warning us that there could be an impending third wave waiting for us in future. And as I've already told, that we don't want to see the pain and suffering what we have experienced in the last few months if there is any third or fourth wave. The question of third wave is coming up because this is happening in different countries across the world. And if you can see in the bottom picture here, the red graph is showing the pattern of waves in US. And as you can see here, the US has already experienced the third wave in the month of January, February, where we had a better time. The green waves are for India, and now we are in the midst of second wave, which is now coming down, and hopefully we won't see the deadly third wave what US has experienced, because at that time there is very little information about the pathology of the virus, the cause of death, the therapeutic modalities, as well as the prevention measurements, including vaccine, what Professor Shah has alluded to you very elegantly. Now from here, so we'd like to face it, and we must face it, and we have to together somehow prevent it. This disastrous fate what we have experienced in the second wave, and probably and possibly the only way to handle and to defend and to fight against these waves is probably we need to inculcate a scientific temper within us. And what we mean by scientific temper, I'll just tell you a little bit of how we usually look at biomedical research and biomedical research data and how we infer. Okay, but as you know, in the public domain, there is a lot of confusion and hesitancy regarding the therapeutics, regarding the pathology, regarding the therapy and the clinical outcome of the patient, as well as about the vaccine measurements, which Principal Sir has also alluded to, that there are some issues with vaccine hesitancy. Now, to inculcate a scientific temper among us, I'll just mention to you about the work of this great personality. His name is Daniel Kahneman. So he was a clinical and experimental psychologist, and he got a Nobel Prize, interestingly, in economics. So he studies basically human behavior. So how we gather information and how we infer. Okay, in real world, that's his experiments. These are his experimental domain, and he has written this beautiful book, very easy to read. It's called Thinking Fast and Slow, and I would emphasize to or press to our student viewers that you might consider reading this book, and I'm sure that it will definitely change your outlook towards the world, how you look at it, and how you'll be looking at it after you read the book. This is an excellent book to read. Now, what he said in the end of the book is that he talked about human, okay, and what is human, and he said, and he has a famous quote called that human is a storytelling animal. Okay, so we love stories, and we confer most of our come to most of our life decisions based on the stories we hear, and we love to hear stories. So what I thought to to think about scientific temper as such, so let me pick up one story from him only. So he has this beautiful story about a thought experiment on about a caveman. Okay, so think about a caveman who had no idea how the external world looks like. So he takes his one child, to the outside the cave, and a big tiger came in front of them, and the tiger pounced and killed his child. Okay, so from this phenomena, this caveman concluded that tiger is a dangerous animal and it could kill your child. So that's a genuine conclusion. So we know that tiger could be really dangerous. So on another day, this same caveman takes his another child to the outside, and what happens? A flock of birds, they flew over the sky, and the child suddenly collapsed and died. Okay, and what he then concluded that, okay, if the birds fly up in the sky, it's also a danger sign and it could kill the child. So we know, So all the conclusions, both the conclusions came from two observations, and in scientific terms we call it correlation. So we correlate two different events and try to make a causal relationship, and that's we do most of the time in our day-to-day life, and that's why it's very important that correlations of different events may not relate to the causality of that particular phenomenon. So, uh, so the point is, so we oftentimes we make these mistakes, and that creates a lot of confusion. And I'll tell you how it happened. And I have talked to a lot of patients and the patient's family, along with my students and other colleagues, and a lot of other people in the last 1.5 years. And what we find that uh, we somehow end up in a matrix like this, and this is about wearing a mask during as a preventive measure. So what people observe that someone is not wearing a mask and he or she never encountered the disease. There's a left top corner, right? But there are people who never wore mask and they contracted disease, the red box. The opposite is also same. So there are examples where there is mask wearing is there, but the patient never developed, the individual never developed COVID, whereas there are some unfortunate people who wore mask and they also developed COVID. And this both positive, this quadrant, right, lower quadrant, is most common among the health workers, doctors, nurses, and other health worker people. They always wear mask, but they also get infected, right? So we basically have all the four observations, right? But still, the scientists and the physicians and the clinicians, epidemiologists, everybody is vouching for wearing mask, right? And they are talking about mostly about this, the green zone here. So if you wear mask, so the question is, it's not that mask wearing will actually prevent disease in 100% cases, right? If you think about this matrix, those who are from biology, you can easily correlate this kind of four matrix with the classic Gregor Mendel's experimental data, right? Where he basically bred tall and dwarf pea plants, and he got all kinds of phenotype, right? And he categorized into their four groups. And from there, he came to a conclusion, right? So here also, so if you look at uh, the percentage of people who are wearing masks and who are not wearing masks, and percentage of patients having the disease, then you can have a better statistical understanding or probabilistic understanding of of the situation, and you can come to a conclusion that wearing of mask can reduce. So wearing of mask can reduce chances of infection. So it's not wearing of mask will prevent infection completely, but it will reduce the chances of infection. So that's the idea. And our problem is that in the public domain particularly, we pick up an isolated event, an isolated experience, like a group of birds flying on top of our head, and the child is dead, right? So that's not the way of looking at scientific data, and particularly in the context of biology or in the biomedical science, it's always, if not 100% times, it's statistical in nature and it's probabilistic in nature. It's not in the yes or no zone. It's always in the middle. So the awareness and understanding in biology is somewhere lies in this probabilistic and determinate continuum. So what we expect from science is mostly a purely deterministic standpoints, that if you take this drug, it will cure the disease, and it never happens. So the point is, if you take a medication, the chance of cure is higher. So that's the better way of putting the real world scenario or the biomedical scenario. But in most of the cases, physics and chemistry particularly, they tend to focus more on the more of purely deterministic terms, where biology is mostly trend towards probabilistic terms. So in probabilistic, the output is independent of input. So that means, so always if your input is 2 plus 2, the output is not necessarily 4 in most of the cases. So that's where the probability comes in. That's where the chances are comes in. And that's what you have to keep in mind when we are talking about any disease, the disease pathology, the disease outcome, the response of a drug, response of a vaccine, response of other preventive measures like mask. Okay, so with this background, so I'd just like to emphasize two other issues that actually creates more confusion and hesitancy is our innate some cognitive bias, what we usually carry. So one is called denominator neglect that we often hear in the public spaces. So here what happens, so you see A and B, in both the cases, people usually pay too much attention to the numerators, right? Oh, I have seen this person got infected. He never had any comorbidity, nothing, but he died. So that means the notion of comorbidity is not applicable for COVID. Okay, so a person can make this comment, but it's because of picking off of certain or small samples and some small phenomena, number of phenomena, and try to come up with a jump to a conclusion. So if your denominator is very large, and then the numerator, the outcome, or in this case it's death, becomes more and more sensible. And if you take like thousands or 2,000, 20,000, 1 lakh patients, and you look at the death records, then you would better appreciate that yes, the presence of comorbidities like diabetes, like hypertension, existing renal pathologies had the patients with all these comorbidities had a higher chances of getting into severe disease and into death. Okay, this is one problem we always face. And another is called availability heuristic, or also called heuristic bias. So this cognitive bias is based on very specific certain informations which are very recent in nature, are very extreme in nature, and vivid in nature, and mostly negative in nature. As I have mentioned, oh, I know someone very young who died from this disease, had no comorbidities. It's the same problem. So here, what people do, they pick and choose, they cherry pick one or two information which is consistent with their pre-existing notion, and they try to fit one or two phenomena with this bias, right? And that's what. So these two examples are also experimentally shown by Dr. Kahneman, and I have taken all these ideas from him, and he actually showed that these biases, whether it's a denominator neglect or a heuristic bias, they actually skews our our conclusion, and which skews us from what's happening in the reality. And we have seen a lot of examples during this COVID days. Okay, so same can be applied to different drugs. So as you can see, we have probably all of us, we have seen a lot of drugs that have been shown or told to us that these are effective against COVID disease, COVID-19 disease, right? And here also, you can also put this matrix where you can show even there is no drug treatment was done, COVID, there was no COVID, or the COVID patients survived without any medication. Even And also, if there is no medication, COVID disease happened. Same with if there is medication, COVID was not cured, and if the medication was given, COVID was cured, right? So all kinds of scenarios are possible, and we see that. And again, our denominator bias, denominator neglect, and heuristic bias forces us to conclude what we want to conclude. Now, as you can see, there are a whole bunch of drugs that were told to us that could be effective against COVID-19. So you see on top, it's a coronal drug, which is Ayurvedic medication. There is on the right bottom, you can see it's a arsenicum album, a homeopathic medication. There is one drug, Zincovit or zinc vitamin combination, was told that could be helpful. And there is an injection called remdesivir, which could also an antiviral drug, which could also cure COVID-19 disease, right? So interestingly, uh, and you could have all kinds of scenarios with all these four drugs, okay? And again, you cannot conclude, because unless you do a rigorous statistical analysis, you won't be able to tell that which drug is effective or partially effective against this disease, okay? And that's why picking up one or two examples from and with our convenience and with our ease will not make us safer. Now, I just like to tell you one story, and again, it's kind of a story of one drug and how modern science is actually takes up its own drugs and criticize it. Okay, so let's see this journey, and I think we will find it interesting. That so in March 2020, so University of Oxford and its institutes, they have a hypothesis that hydroxychloroquine and chloroquine could be a potential drug to treat COVID-19 disease, right? And you see, it was proposed in March 2020. So the researchers across the globe, they took this challenge, they tried to study it, and excuse me, and most of the studies were done in vitro cell culture where coronavirus was infected and plaque formation was studied. And there's a very nice, interesting data came up by June 2020 that hydroxychloroquine indeed can actually prevent coronavirus infection in the cell culture model. Okay, and these studies were done in a cell type called verocells, which is for biology student, you might appreciate that these cells are from this is a monkey cell and it's a kidney cell. Okay, so the kidney cells, if kidney cells are treated with hydroxychloroquine, these cells are protected from coronavirus infection. And with this, there's a lot of hope that this medicine could be of help to us. But the modern medicine doesn't stop here. Okay, so people start asking questions based on these data, and they found they started embarking on a lot of other experiments. And interestingly, by September 2020, you could see a paper in Nature came up where they have shown that the hydroxychloroquine use against SARS-CoV-2 in non-human primates. Okay, so in the monkeys, and what they have shown that hydroxychloroquine has no effect of preventing coronavirus infection in monkeys. Okay, and particularly in the uh, in the in the lung epithelial cells. Okay, so it could prevent infection in the kidney, but it cannot prevent infection in lung, where is the primary site of coronavirus infection. So this was a big setback for hydroxychloroquine, and as you know, there's a big hue and cry on hydroxychloroquine therapy, its preventive prophylactic measurements people have started taking, and these questions, but although it was in practice, but scientists were still questioning and trying to come up with new experimental evidence to suggest that, well, this is not the last word, and it has to be questioned. And science, the modern science particularly, has this beautiful internal checks and balances where a questioning is allowed and new experimental evidence are allowed to publish and to defend or refute the previous claims. Okay, and by April 2021, now we have a huge metadata analysis, which is nothing but there has been a lot of clinical trials involving patients with hydroxychloroquine and chloroquine. All the data were put together and rigorous statistical analysis was done. And what they have shown by April 2021, that mortality outcomes with hydroxychloroquine was minimal, and if not, hydroxychloroquine therapy can actually increase, interestingly, it can actually increase chances of mortality or chances of death in severely infected patients. Okay, and as you know, by this time, so hydroxychloroquine is out of business in the context of COVID-19 therapy. So, why I am doing all these exercise and showing you how modern medicine works. Unfortunately, you won't see that in either in the coronal kit or this Ayurvedic medicine. We don't have this internal critique, or in homeopathic medicines where we don't see this internal critique and where one is questioned, one's data, one's claim is always questioned by their peers, and we call it peer. So the friends among us, among the scientists, we question each other, and through questions we enrich each other. Okay, and that mechanism is unfortunately absent in other modes of medical practices, and I hope this practice will come up in other fields also, and we'll have a better world in that sense. And for this questioning and to study the effectiveness of a particular drug or a particular preventive measure, vaccine or anything else, the best possible method is called randomized controlled trial. So what it is done, so a group of patients are taken and they are statistically randomized. Okay, so there is a statistical method by which there two groups are formed. Okay, and one group is given treatment, one group is not given treatment, although they don't know the group, they don't know that they are given treatment. Okay, so we call them a placebo group or control group, and then these patients are followed up, okay, after giving a treatment or vaccination. Okay, and the outcome is measured. Here you can see here in the follow-up group of the treatment cohort, you see only one patient is not cured, whereas in control group you have 3 in this example here. And then you can compare the results and you can tell, okay, so this method, whether it's a hydroxychloroquine or a vaccine, could be effective or may not be effective. Okay, so that's what is called randomized controlled trial, and this is done across the globe. It is done here also, and some of my friends has also done RCT on plasma therapy in Calcutta, at the ID hospital also, and so this is the best method till now we have, and this is not the end of the world. There could be much better method which can come in future, because our peers are always questioning the methods, and we might see a better method of analyzing the effectiveness of a particular medication. Okay, but with all these efforts, unfortunately, we still don't have any specific antiviral drug against SARS-CoV-2. Okay, and we should keep that in mind. And modern medicine is open to this idea that it has claimed that there will be a whole bunch of drugs which could be helpful and can be a potential antiviral candidate. But unfortunately, even after 1.5 years of this disease, we have no specific antiviral drug. Okay, so a lot of research are going on, and scientists are trying to come up with new antivirals. Okay, okay. So now, if we don't have a very specific medication, then what are our options? Okay, So options had what Professor Shaha has already mentioned. So the app option is basically vaccination. So and develop a protective immune response in us, right? And we can get it through two different modes. One is natural infection. When one person get infected, he or she will develop a protective response in terms of antibodies, memory T or B cells, and which can give a protection for a few months or a year. But natural infection, so the idea is that then you have to allow entire population to get infected so that we don't see these waves of infection in the future days. But natural infection comes with a caveat that at least 5 to 10% of these infected individuals will develop severe disease. That means they will require oxygen therapy, hospitalization, ICU therapy, ventilation, ECMO therapy, and all kinds of therapy modules. And there will be 1 to 1.5% unfortunate individuals who will succumb to that disease. So natural infection will not be the ideal way of looking at it. Because if we think about Indian context, we have 130 crore people, and if we are talking about 1 to 1.5% death, so it comes out around more than 2 to 3 crore individuals. So we cannot allow that. Okay, ethically we should not allow or allow death of 3 crore individuals, 3 crore Indians in few years from this disease. So the best option we have basically these vaccinations. Okay, and we have a lot of vaccines, and again there are a lot of hesitancy and confusion about vaccines. But if we look at just the what is the state of natural infection, so we embarked a study across 40 different centers in India. And I was part of this study. It's called Phenom India cohort. Here we looked at what was the seroconversion rate. That means what is the antibody levels of individuals across the country. Okay. And what we have found by the end of September 2020, it was around 10%. Okay. Whereas by end of December, January, it went up to 15 to 20%. Okay. So the antibody response, so the antibody titer was only 10-15% in the across the country, which is very, very low compared to the herd immunity, what Professor Shah has already alluded to. So we need to, so apart from this, so after the second wave, this percentage will still go up, maybe 20% to 30-40% in some areas. It will be 50 to 70%. But most of the countries will be still under 30% range. So, and interestingly, these antibodies are not lifelong protection. We have seen that antibody levels actually go down from within 3 to 6 months of time. So this natural infection also does not give you a long-term protection. Okay. So then we are left with option of vaccine and mass vaccination, because so the data are coming across the globe on different vaccine candidates, and this is a very recent data published in Lancet in month of June, and I have only emphasizing on the data of AstraZeneca Oxford vaccine, which is known as Covishield in India. Here you see the red bar on top. It's called relative risk reduction. It's 67%. That means if a person takes the vaccine dose, 2 doses of vaccine, that person will have 67% reduced risk of developing the infection compared to a non-vaccinated person. Okay, so it gives some hope. And also, but the problem is with its absolute risk reduction, which is low, and that's why another term is called NNV. So the number needed to vaccinate for people to prevent one more cases of COVID-19 is 78. So that means if you want to prevent one new case, you have to vaccinate at least 78 individuals. As Professor Shah has already shown, that if you surround a susceptible individual with vaccinated or immunized people, natural or artificial, you need at least 78 individuals for Covishield, right? So that means we need a massive vaccination drive across the country to prevent future waves of coronavirus pandemic, right? So here, another recent data, it's published in June 2nd in 2020, this month only, published in Nature, and what it shown that almost 170 crore people has already been vaccinated, at least one vaccine across the globe, and among the different countries here, as you can see here, from Israel to India. So where Israel has already fully vaccinated their 60% of their residents, of their citizens, whereas India, we are still lagging much behind. We have vaccinated 3.2% fully vaccinated individuals in the country. So we need to increase it. We have 20 to 30% naturally infected. We have only 3 to 4% vaccinated individual. So we are not in a good shape to completely prevent a possible third wave. So we expect that if a third wave comes in near future, it will not be as devastating as what we have seen in the second. And with this, I would like to conclude that we still need to long way to go. And from here, I'll be happy to take questions. Thank you.
Thank you, Partho. You gave another dimension of this webinar series, and it is really very exciting. And of course, sad to know that we are still behind. And Dr. Indranil Chakraborty, please. Thank you, Partho.
Thank you.
Thank you, Dr. Partha Chakraborty, for your wonderful lecture. And thanks to Professor Chitra Mandal for introducing both the speakers and taking the trouble to do all these things, which we are really thankful to you, ma'am. Now, I would like to take the honor to request Dr. Amit Ghosh, former Director CSIR, IMTech Chandigarh, to deliver the concluding remarks.
Amita.
Dr. Ghosh, are you there?
Where is Dr. Ghosh? কি হলো? Where are you? হ্যাঁ
এই রে অমিত ফোন করেছিল আমাকে, আমি তো ধরিনি।
You have to stop the entire thing. Okay? Just stop and go out and then come back again.
হ্যাঁ, আমার যেটা হয়েছিল ডিসকানেক্টেড হয়ে গেছে।
না ওই লিঙ্কটা তো থাকা উচিত। আচ্ছা ইন্দ্রনীল,
হ্যাঁ বলুন স্যার।
ডক্টর ঘোষের he has been linked out. তোমরা নতুন করে একটা লিঙ্ক পাঠাও ভাই।
ওকে ওকে।
ওই সেম লিঙ্কটা হ্যাঁ।
ওকে স্যার, I think Shubhroda is sending the link.
সেটা ইয়ের জন্য হচ্ছে, আমার হয়েছিল, এটা নেটওয়ার্কের জন্য হয়েছে। হ্যাঁ সেই জন্য বলছি ইউ জাস্ট ক্লোজ ইট, আর ওরা লিঙ্ক পাঠাচ্ছে আপনাকে 2-3 মিনিটসের মধ্যে, দেন ইউ ওপেন।
আমি পাঠিয়ে দিয়েছি।
আচ্ছা আপনি দেখুন, আপনি স্টপ করুন, then you again come.
লিভ করে আবার জয়েন করুন।
শিক্ষা মন্ডল has already sent you. ঠিক আছে? We are waiting for you for 5 minutes.
আমি অলরেডি পাঠিয়ে দিয়েছি।
ঠিক আছে, ঠিক আছে। He will join. He will join in 2-3 minutes, 5 minutes. The same thing happened to me.
কোশ্চেন আনসারটা কি আপনারা শুরু করবেন, তারপরে অমিতা জয়েন করবে?
Okay, if you
I-
permit, then
I
we
think
can
so.
start.
হ্যাঁ, কোশ্চেন
So I
আনসার
can-
you can start, then he will
start, yeah.
Okay. We have already collected some questions both from YouTube chat box and from Google Meet chat box. Here the question is to Dr. Shah. The question is from Professor Amitav Datta Mojumdar, ex-head department of chemistry of our college. Is booster dose of Covaxin or Covishield required after 2 normal doses?
Well, it depends on what the vaccine makers would recommend. One first dose followed by one booster is what we are seeing now, and the immunity stays, according to reports, is about 6 to 7 months after the second dose. So whether a third booster is needed or not, it's not clearly spelled out as yet.
Okay, thank you, ma'am. I think Professor Mojumdar has got his answer.
So
Here is
can I
another
come in
question.
now? Or
Here
you
is
have the question first, or can I come in now?
Just 1 second. Dr. Amit Ghosh is ready. He is here. So
Yeah.
should we continue this question answer, or I think we should request Dr. Ghosh to come back?
I think Dr. Amit Ghosh should conclude.
Okay.
Okay.
Dr. Ghosh, you
All
please
right,
you please start.
Okay. Actually,
You
after this,
start.
can you hear me now? Can you hear me?
Yes, we can hear you.
Okay. After these two very elegant and erudite presentations, I really have very little to say, but still I'll have some observations to make. First of all, one general observation is, I think we should have more such lectures, because when you look at the television, you find that there are artists, there are film actors, there are cardiologists, of course there are doctors who come later, but seldom any scientists. So a lot of half-baked information goes around, and people who are not into science, like here I see people, students from history and other things, they do get what I would rather say very skewed view of the whole thing. Having said that, I'd like to say that I'm very glad that Professor Chakraborty has mentioned Kahneman's book. Although he said it's very easy reading, I didn't find it very easy reading. It's a very thick book, but it's a wonderful one, because if one goes painstakingly through it, one really understands the importance of the data, and which we are now facing, because when you predict a wave, you need data. And unfortunately, all the predictions are now based on very inadequate data, because for whatever reason, the maybe it's deliberate, may not be it's deliberate, because my experience, I beg your pardon to say this, with many students, that we are not very good at keeping data. The laboratory notebooks show that. So when they say that the government of India is not giving correct data, it may be because the different centers are not keeping proper track. And unless we have proper data, for which a group of 100 scientists from India actually made a presentation to the central ministry, the prediction of the waves will be very difficult. Now, there's just one more point I'd like to make out, that the title of the webinar was "How long we have to fight it?" I believe that it's a very tough question to answer, because even though Professor Chakraborty said that we have covered, and Fauci says that you need 90% infection to have herd immunity. See, there are countries in Africa which are totally out of the loop. For example, a country like Malawi, they have no vaccination at all. Whatever vaccine went there to the Gates Foundation, they got all wasted, because there's no infrastructure. And in today's world, we really don't live in island universes. In ancient times, people traveled by boat. So if one had infection, it showed up during the journey, and so when they reached the destination, they was quarantined. But today it's not so. One can fly in from one continent to the other in a matter of hours, and with inadequate testing and other things, if I am a COVID patient, then I go to a place and I spread it. So therefore, the question of how one is going to be controlled is a very tough question, because whenever there is inadequate vaccination, there is pressure on the virus to mutate. It's a natural law, and therefore this whole business of how do you control it, it becomes a major problem. Even if you look at our country, recently Supreme Court has said that there is a digital divide. Now we are very comfortable with the computers and we can do this co-win and whatnot. But imagine a person in a village where they may not have access to these things. So how many people are going out of this net, and what is going to be done about that? And therefore, you know, with the limited thing, as has been rightly pointed out by both the speakers, that our only hope is to do very aggressive vaccination. But even if we are able to do very aggressive vaccination, still there are countries in the world, if they are out of this so-called this net of protection, we are still not safe. But as I said, the scientists have a way of surmounting the problems. I don't know how many of you know about it or not. Recently, they have discovered 2 coronaviruses, one from the pig, which is in Haiti, and the other in Malaysia. And this Malaysian coronavirus have come from the dogs. They have not yet jumped the species, but scientists are really scared that if they jump species, then what is going to happen? Then you have one more set of things to worry about. So these just the observations I thought I would make, because the webinar title is how long we have to fight it. It looks to me the fight is a long one, but as I say now, there's a Black American poet, Langston Hughes, he said that, never cease to dream, because when you stop dreaming, the life is like a barren field full of snow. So with that, I'll stop my comment, and then we can have the questions.
Okay, thank you, Dr. Ghosh, for your valuable remarks, and the way you summarized the things are really praiseworthy. I once again thank you very much from the core of our heart. Now we shall go back to the question answer session. I have asked only one question, and there are questions in queue. Another question to Professor Shah is that this question is from Sunil Mukherjee. He asks, another antiviral adaptive pathway within bracket RNA also contributes to eliminate viruses as well as other pathogen. Would you make any comment on this? Should I repeat, ma'am, question?
Yeah.
Another antiviral adaptive pathway within bracket RNA also contributes to eliminate viruses as well as other pathogen. Would you make any comment on this?
Yes, it is known that this pathway works, but since I am not an expert on the RNA biology, I will not be able to make comment on this.
Okay. Then the next question is from a student, Riya Sharma. She wants to know, is Covishield AstraZeneca a vaccine?
Yes. Covishield is a vaccine. AstraZeneca, it was based on AstraZeneca vaccine. It is made in India. It's a vaccine, yes.
Then from Anupama Patnaik, a teacher of physiology department, she asks, respected ma'am, there are so many processes to activate immunity against SARS-CoV-2 virus, but which process is more effective?
There is activate immunity is through vaccines only, that one can activate the immunity to the virus. And see, there is a lot of variability as per how immunity will work against a virus or how they will respond to vaccines due to the presence of something which is called a MHC in our body. And each person has a different set of MHC, and that is why the vaccine response is so variable. So it is difficult to say which is the best one, but the only way you can activate your immunity real good is through vaccination only.
Thank you, ma'am, for answering the questions. Now, here are some questions to Dr. Partho Chakraborty. This question is from Dr. Shukla Mandal Shah, the respected morning in charge of Kharagpur College. I am quoting her. Respected sir, correlation and regression states the relationship between the concerned variables. On the other hand, causality test can give actual causes. Is it tested, sir?
Yeah. So yeah.
Indonic, not
Hello.
it is causality.
Okay, ma'am, you may ask the questions directly. You ask the question.
Actually, sir, then, that there is a those available data correlation regression among the different variables, and there find causal among these different variables. Situation I that question that is causality
Kindly switch off your microphones other than the speaker. There's a lot of background noise coming in.
causality test among these variables. My question. If causality, and then we can find of this variable which is cause and which is not.
right, right, absolutely. I mean, you are absolutely correct that correlation and regression will only give you the strength of their association. It will not give you a direct causality per se. So that's why you need to do experiments.
Yes.
So in experimental setup only you can talk about causality. And
Yes.
you eliminate one variable and see the effect, and that kind of experiments you can do. And in the context of coronavirus, lot of experiments with real virus has already been done. And so now we have good experimental evidence to say that, well, this virus works this way. This spike protein binds to ACE2 receptor with these residues with almost atomic at the atomic resolution level. So, and how virus enters the cell, how it infects, how it induces the inflammatory response, cytokine storm, all these pathologies has all have been delineated at the experimental level. So we are pretty sure now that yes, there is a causal relationship between these two.
Okay. Thank you. Thank you. It is time needed factor.
Absolutely, absolutely.
Yes, sir.
Yeah.
Okay. The next question is from Professor Mahanga Singh of Department of Botany from our college. Is it safe for person having autoimmune disorder to take vaccines?
Okay. So it depends upon what kind of autoimmune disorder we are talking about. If it's a organ specific, like hypothyroidism, type 1 diabetes, so there is no concern. One can easily take the vaccine. But for systemic autoimmune disease like psoriasis, SLE, rheumatoid arthritis, like that, and if you are under some immunosuppression, so whether it's a steroid or methotrexate or other drugs, then you have to talk to your concerned physician, look at your clinical status. So immunosuppressive might have to stop for some days. All those people are still there are no clear-cut data, but it has been shown that even in the presence of steroid medication, vaccines do work, although the response is not that great. So I just would like to add to this, which I missed in the presentation, that the immune response is also severely compromised, statistically compromised significantly in the older patients, older individuals. So that we also keep it in mind. So when we are talking about mass vaccination and particularly vaccinating the susceptible individuals. But for autoimmune disease, that's my take. Yeah.
Okay. Another question to you, sir. It is that is the possibility of cytokine storm higher in case of a strong immune person?
So, well, the best way to put it, it's actually stochastic in nature. So, strong immune person actually means nothing in terms of his or her immune repertoire. As Professor Shah had shown, the involvement of so many cells and their complex interactions. So what we mean by strong or weak immunity, and that is not very clear. Okay, that's the first thing. And also the cytokine storm is particularly happens, as you know, oftentimes after the virus leaves the body. Okay, so it's a post-viral phenomena in a lot of cases. So in clinical observations, we have seen all the severity happens in the second week. Almost 90% of the patient, everything, all the bad things happens in the second week. So it's very, it's not predictable who will develop severe disease or cytokine storm.
Okay, thank you, sir. Here is one more question from my personal behalf. Actually, we have learned, we have heard that the viral diseases cannot be treated with antibiotics, but we have seen that azithromycin and doxycycline, these are antibiotics. Why these are used to treat SARS-CoV-2?
Absolutely. So again, so what happened in the initial days of this COVID-19 research, people used to study in this virus in the vero cells, as I said. In the cells, they look at the infectivity. And what scientists did, they took all the available drugs in their hand. Okay. So idea is to repurpose existing drug against SARS-CoV-2. That would be the easiest thing to do. And they used all possible, almost a million, chemical compounds were used to see which ones actually can inhibit viral replication. And interestingly, doxycycline and azithromycin showed some inhibitory actions. So we know they are antimicrobial, antibacterial, right? But they, at least in vitro cell culture system, they showed some antiviral impact. And even there are some data to show that even hydroxychloroquine and azithromycin has a synergistic effect. So those data are also available. So that's even including ivermectin, which is an anti-parasitic drug. So these all drugs, they showed in vitro viral inhibition of viral replication. But time, as you know, with other clinical trials and data, nothing actually stand the challenge of time.
Okay, sir.
Yes,
Thank
I
you
have
very much.
one more question. That after vaccination, someone is
Yeah.
affected by fever and others, so many others problem, pain, so many huge problem, and some are not
Right.
affecting anything, just like
Right. Absolutely.
a few pain on the side of this vaccination. What is the reason?
Well, again, as you said, I mean, so the immune diversity is it's so diverse. I mean, because we are all different, right? We are genetically different, physiologically, in every possible way we are different. So our immune system, which is a very complex repertoire of cells and cytokines, chemokines, and everything. Okay, so it's so different. So we would definitely expect a differential response to vaccination. So we'll definitely see some people will not have anything in one side of the spectrum. On the other side, we'll see really strong side effects, including, as you know, there are incidents of clotting with Covishield vaccination.
Yes,
Okay.
yes.
Right. So you'll have the entire spectrum because that represents the entire humanity and their immune diversity.
That which is those are not
I,
affecting more immune power, and those are not affecting.
Not that the case. That's not correlated with their immune power.
Yes. Okay, okay.
I,
Thank you very
I,
much.
I have a point. Can I make this point before you?
To me, yes,
Yeah,
please.
Dr. Partha Chakraborty.
Yeah, please, Antada, please.
See, recently, many of us, including myself, I got an email it's from the government of West Bengal. There is a notice, and it's a circular from West Bengal that we are waiting for Madhyamik, Uच्च Madhyamik, ICSE, CBSE examinations. Examinations cannot be conducted at this stage. And now, after hearing your lecture and Dr. Chunyosa's lecture, I already sent my opinion to the government, and I agreed, and my opinion is in favor of Dr. Amit Ghosh's statement, because the vaccination should be aggressive, very, very aggressive. If you start vaccination to the candidates, there is no point that 18 years old below, they should not be vaccinated, because we are very much concerned about the old age people. But now this is the time where everybody should be vaccinated. Completion of this vaccination should be done. So if you start vaccinating all the students, 23 lakhs of students are there who will appear for the examination. And if you vaccinate 23 lakhs of people, of the students, it may take 1 month. So if you start from today, this vaccination for 1 month, and then wait for another 1 or 2 months to activate this antibody preparation, the whole thing will take only 3 months. Now you can take the whole examination after 3 months. Then in that case, you will not lose much. 3 months after you take the, because you are planning to take the exam in July. So if you start this, it will, the whole period will be completed in three months, and then you can take the examination. You don't have to wait for this social distancing and other things. That you can conduct the examination in your home, in your schools and convenient places. That is my suggestion. That is my suggestion. I have already sent to the government.
May I make a small comment here?
Dr. Chakraborty, can you start your vote of thanks and next program, because we are already one day.
No, Chitra, may I make one comment here? Will I be allowed to do so? I just have one comment to make. Can I make that comment?
Yes, Amit.
See, Hemanto, the problem is the number of vaccines available. You know, there's a Nature article which showed that out of 8.6 billion doses that was created, more than 6 billion were cornered by high income and middle high income countries. So the question is, if really 1.2 billion Indians have to be vaccinated, the vaccine supply has to be there, which is not there. That is the main problem, apart from other political and other considerations.
No, this is selectively for the students who will appear for the examination. That is my point. Take a workful practice strategy and vaccinate the candidates. Then you take your time to vaccinate the entire population. That is the suggestion I have made.
Okay, now we shall proceed. Shubhru, can you start the next event?
Okay, thanks to all the speakers. Now, I request Dr. Shubhru Mishra, the convener of the webinar organizing committee, to kindly announce the names of the top 3 rank holders in the student seminar competition that was held on 2nd and 3rd June of this month. Okay, Dr. Mishra, please.
Oh, Shubhru, where's Shubhru?
Shubhru is not there. Can you call or something?
Microphone is switched off, muted.
Do you have the name?
শুভ্রর মাইক্রোফোনটা আচ্ছা জয়েন করেছে।
আচ্ছা, করো।
শুভ্র,
Yeah.
শুভ্র স্টার্ট।
Subhrata, unmute your microphone and announce the results.
Hello.
Yeah.
না একটু
Am I
ফোন করে
audible?
বলতে হবে, ও তো আনমিউট নেই, মানে ও তো শুনতে পাচ্ছে না।
Hello.
প্লিজ আনমিউট, শুভ্র প্লিজ আনমিউট,
Am I audible?
আনমিউট করুন।
Okay, madam. Good afternoon, everybody. It's an honor and privilege for congratulate the winners along with all those who have taken part in the second day's online, 2 days online seminar competition on waste management and renewable energy harvesting to restore the earth.
আনমিউট প্লিজ।
Unmute to us.
His
Unmute.
microphone is
ম্যাম,
on.
আমার মনে হচ্ছে ওর ওখানে সিগন্যাল খুব কম। আই হ্যাভ দ্য রেজাল্ট।
অন্য কেউ রেজাল্টটা বলে দিলে ভালো হয়।
হ্যাঁ, আমি তাহলে বলে দিচ্ছি।
বলে দিন, ওনার কানেকশনটা
Unmute করা আছে? শোনা যাচ্ছে?
না না, এখন শোনা যাচ্ছে, ক্লিয়ার শোনা যাচ্ছে না।
জ্যোতির্ময়দা বলে দাও। না, জ্যোতির্ময়দা বলে দিক।
So on behalf of Subhro Misro, Dr. Subhro Misro, I am announcing the results of the 3 rank holders on the student seminar competition that was held on 2nd and 3rd June. First position secured by Sri Deepak Bera, 6th semester students of physics department, and he presented on renewable energy harvesting. He secured 35.8 marks out of 50 marks. 35.8 out of 50 marks. There were 5 judges, so we mean there are 5 judges marks, and it is 35.8. And second position secured by Sri Semapadopal, a 6th semester student of physics department. He talks on waste management. He got 32.4 marks out of 50 marks. And third position secured by Sri Abhijit Rai, a 2nd semester student of zoology department. He got 31.6 marks out of 50 marks. And there are total 30 participants, 30 students present their presentation, among which they are the first, second, third. So we can congratulate all the 3 students for their nice presentation.
Okay.
And
Okay. Thank you, Dr. Pramanik, for your announcement. Now we have come to the last part of the program. I would now request Dr. Jyotirmoy Pramanik, Joint Secretary of the Webinar Organizing Committee, to propose formal vote of thanks.
So good afternoon all. On behalf of organizing committee, I take this opportunity to propose vote of thanks to those who have directly and indirectly contributed to this national webinar. We are really honored to get this opportunity to conduct this webinar jointly with such esteemed association, Indian National Science Academy, Kolkata chapter. First of all, I wholeheartedly thank eminent scientist Professor Hemanto Kumar Mojumdar, Senior Scientist Platinum Jubilee Fellow, CSIR IICB, Convener INSA Kolkata chapter, for his constant guidance during organizing the webinar. I also like to thank Dr. Chitra Mondol, ACRB Distinguished Scientist, CSIR IICB, member of the Executive Committee, INSA Kolkata chapter, for agreed to conduct this webinar in collaboration with our college and provide all kinds of supports. We are very much grateful to Dr. Chandrima Saha, President of Indian National Science Academy, former Director, National Institute of Immunology, New Delhi, for being with us as a keynote speaker. We are really thankful to her for managing her time from her busy schedule. I would also like to thank Dr. Partha Chakraborty, Principal Scientist, Cell Biology and Physical Division, CSIR, Indian Institute of Chemical Biology, Kolkata, and Dr. Amit Bose, ex-Director, CSIR IM Tech, Chandigarh, JC Bose Distinguished Chair Professor of NASI, ICMR, NICED, Kolkata, for enlightening us with their knowledge and gave deep insights into the topic and also revealed some interesting facts. I must thank Sri Sondip Sen, DSC member, HS Council, for his continuous support during conduction of webinar as well as the students' competition. We are grateful to our beloved principal for his encouragement and support to organize this webinar. I like to convey my special thanks to the judges, Mr. Sandeep Sen, Dr. Bimal Krishna Das, Associate Professor, Department of Mathematics, Professor Mahanga Singh, Associate Professor, Department of Botany, Professor Devashish Aich, Assistant Professor, Department of Physics, and Professor Prasanna Dule, Assistant Professor, Department of Chemistry of Kharagpur College, for evaluation of the oral presentation in the student seminar. Also, thanks to all the members of the organizing committee and advisory committee for their help and suggestions. Also, thank you all to all the participants present here for paying your attention and learning. I would end my speech here. Thank you.
Now, ma'am, I announce that the webinar is over here. Okay. Okay. We hope to meet once again very soon. Thank you all.
Thank you. Thank you.
Thank you, sir.
লাইভ স্ট্রিমিংটা বন্ধ করে দিতে হবে।